Hello everyone and welcome to today's live broadcast, Early Disease Detection Performing NMR, Understanding How a Better Analysis of Metabolites in Serum and Urine Can Help. Presented by Professor Claudio Luchinat and Professor Elaine Holmes. Today's presentation will be moderated by Manfred Sproul. I'm Christy Jewell of LabRoots and I'll be producing today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Bruker. Bruker enables scientists to make breakthrough discoveries and develop new applications that improve the quality of human life. Bruker's high-performance scientific instruments and high-value analytical and diagnostic solutions enable scientists to explore life and materials at molecular, cellular, and microscopic levels. For more information, please visit bruker.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click the Send button. We'll answer as many questions as we have time for at the end of the presentation. We will additionally answer them offline after the webinar. If you have trouble seeing or hearing this presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. Lastly, near the end of the presentation, a poll will pop up on your screen, which we kindly ask you to complete. Your answers and suggestions will help improve our webinar program and is greatly appreciated. It is now time to enter the topic of the day. Manfred, over to you.
Thank you, Christy, for this nice introduction, and thank you everyone for joining us today. Our webinar titled Early Disease Detection Performing NMR, Understanding How a Better Analysis of Metabolites in Serum and Urine Can Help, is actually presented now by Claudio Luchinat and Elaine Holmes. Professor Claudio Luchinat is a full professor at chemistry at the University of Florence. He is a co-founder and director of CERMMP, the Center of Magnetic Resonance, and the CIRMMP, Interuniversity Consortium on Magnetic Resonance of MetalloProteins. His research interests include development of NMR-based structural methodologies, electron and nuclear relaxation, NMR of paramagnetic species, relaxometry, bioinorganic chemistry, and what we are talking about today, metabolomics. Professor Elaine Holmes is a professor of chemical biology and the head of the Division of Computational and Systems Medicine at Imperial College with over 20 years of experience in metabolic research. Now, first of all, I would like to give a short introduction to show the basis where this research actually is done on, and this is on our IVDR platform that is used in many of these metabolomics research labs. So here we are actually talking about a platform and a concept overall. IVDR stands for In Vitro Diagnostic Research. It is a completely standardized, fully automated concept using this 600 megahertz platform with predefined hardware and also embedded solutions for clinical and translational research. The big advantage of this concept and the platform is that we can integrate of results from different users all over the world. Of course, that creates a large big data pool that can be used for much better statistical analysis and using data from all over the world. And this is due to the very, very good reproducibility of NMR and also the transferability of NMR. So also, of course, having such a large data pool from different sources, it can help to really speed up the translation into future diagnostic methods. And that could be either for personalized medicine, which is a very important topic these days, as well as for big studies on population basis to look, for example, into phenotypes and associated risk factors. And here I want to shortly introduce this platform and the enabling tools. So here you can see now the instrument or the platform in the middle. This is a 600 megahertz based on a 600 megahertz magnet, which actually has a very good shielding. So that means there is no danger outside of the magnet for any magnetic disturbances that would be there. And what we are working on, I start on the top. This is, for example, Phenome, where we have several important collaborations. We are working with the Phenome centers. You probably be aware of the International Phenome Center network that has been brought to life about 1.5 years ago. And this now includes centers which are located in Europe, like in London, in Birmingham, and in Singapore, also in Australia, in Perth. And of course, there are quite a few groups to join this International Phenome Center network. And using the standardization that NMR provides here, they can actually exchange all their data. We also work on a biobank QC program that has just been released, which allows actually to check the quality of specimens which are in the biobank, but at the same time to generate spectra and also analytical data like quantification on those spectra that can be stored into a biobank. But we also talk to clinical service providers, hospitals, and medical practices. This is more for the future. But also work on forensic departments. Environment and human health is an important aspect here. And of course, we also have applications for pharma industry, just to mention toxicity screening, dose response optimization, and of course to look into metabolite ID. That means the metabolites formed from drugs in the body. Now, the concept is the following. You see here the basis. This is this dedicated platform at 600 megahertz. This is under ISO 9001 and ISO 13485. On top of that, we have what we call our IVDR methods, the SOPs, which describe how to collect samples, how to store samples, how to prepare samples, how to measure samples, and also a standardized analytical procedure to follow. In addition, there is a spectral compound reference spectral base of compounds occurring in body fluids mainly that has 800 compounds in there with more than 23,500 spectra included. And also there is a forum for the users of this instrument platform to exchange information with us and of course between other users. On top of that, we have the layer which we call the enabling tools. And so the ones which are available in the moment is a lipoprotein subclass analysis. We have plasma serum quantification of small molecules that has just been released. We have a neonate urine quantification with 150 compounds which are quantified under full automation. We also have child-adult urine quantification, also 150 compounds. And also released already is the biobank quality control tool. On top of that, we have our R&D ecosystem, where actually customers of ours or collaboration partners of ours that are using this platform and the SOPs, they are working on development of diagnostic routines, which then, of course, can be put onto this platform and made available to other users. You see here an iPad because this is the concept that we are following. We have a fixed hardware platform and we have a fixed interface definition, how applications are actually talking to this whole system. And therefore, of course, we have here really the same concept to be able to actually have multiple solutions on an instrument to make sure that this instrument can work very efficiently. So what does that mean? You can see here, for example, this is a little bit an analogy to the internet. So in the internet, of course, there are protocols which have to be kept. Otherwise, Google, Amazon, and how all they are called, they could not work. And on the same thing, like the Internet protocol, we have our SOPs, which make sure that users can exchange data between themselves, but also with Bruker, for example, for a remote analysis. Now, starting a little bit in an explanation, what are the enabling tools? So if we look, for example, on the SOPs for serum and plasma, when that is used, then we can deliver the quantification of, for example, here BI-LISA. This is our lipoprotein subclass, and the quantification of small molecules. We have made sure that this can run on a fast experiment that only takes about 8 minutes, give us lipoprotein information. This is about 115 parameters, and also 26 small molecule quantifications from exactly one same experiment. And then, of course, there is also an extended version which has some additional experiments which allows us to do even more sophisticated quantification and analysis. And then we have the Biobank QC tool, which works also on the SOPs that we are using here for the other analysis. Now, when we look into quantification on small molecules, this is important in epidemiology. A lot of health problems, cardiovascular disease, diabetes, cancer. Then, of course, we can have information about personalized profile in plasma and serum and identifying the metabolites which are relevant in there. We can also look to food-related changes in plasma serum composition to show the influence on health. And of course, it allows us, for example, to monitor and optimize treatment. Now I just show you a report. This is the small molecule quantification. So you can see here the components, and you can see that we have a large variety of chemical classes that are actually analyzed with one single experiment, which conventionally might take actually more than 1 or 2 methods. And you also see here, when we look into this gray bar, that this is a normal concentration that we get from our reference cohorts. And the black bar, this is actually the person itself, so you always know if that person is within the reference range from our models or not. So it's explained a little bit better here. You see here in gray, this is a 95% range in the model set, and then the actual person, which in this case here really is inside the range. If you look down here, you can see, for example, here we look at pyruvic acid, which is clearly outside of this normal model range that we have identified from a large number of compounds. So the quantification actually is quite complex, but you don't have to care about that because this is all done in automation. So you can see how these signals are identified. You can see here, for example, creatine or creatinine, which are somewhat overlapping, but the program takes care of this without having any problems. Now, for example, when we look into TMAO, trimethylamine oxide, of course, this is a relevant metabolite also when we look into cardiovascular health. And here we see, for example, how you can look from 2 different angles to the same disease. And we have the angle of the small molecules, as you can see it here, and we have the angle actually of the lipoproteins, which of course also play a large role, like when we look into food, especially food and health, high blood pressure, fatty liver disease, then of course inflammatory cancer, cardiovascular, and several others. So how does that look? What you see here now, this is a report, again, looks quite similar to the small molecule report you have seen. So we have these gray bars determining the normal ranges in our model, and then you have the actual person, which is identified with the black bar. And as I mentioned before, there are main parameters. These are the main, the overall triglyceride cholesterol, and then we go into the main classes: LDL cholesterol, HDL cholesterol, ApoA1, ApoA2, and ApoB. We have particle numbers, I will not go into detail on all of that. I just want to show you a comparison when we look into a control sample and a donor which actually had a cardiovascular event 3 days after the blood collection. So if we look here on the control sample side, you can see that all of the subfractions, whether it's triglyceride or cholesterol, free cholesterol and phospholipids, the person is in the reference range. If we look on the right side, you can see that there are substantial deviations from the reference range. And what is also important, for example, when we look into the LDL, you can see in the LDL main fraction, the person here is actually quite in the middle of the distribution in our reference cohort. But if you look to the individual subclasses, you can see that this distribution actually is quite unhealthy, because we have the health actually is improving when we have here a high concentration on the first 3, LDL 1 to 3, and a low concentration on LDL 4 to 6. And here it's vice versa. But if you just take your doctor's main panel that you would get from your house doctor, he would not recognize that there is a problem. NMR can do that within a few minutes. Good. We have, of course, done very relevant now studies, a ring test to see how transferable these results are. So you see here a comparison of the results on a NIST standard sample for 11 instruments at 5 different sites. And you can see the R value here of the correlation between the different instruments, and the average is extremely good. And the last I want to talk about, this is the quantification in urine, which of course also supports, for example, epidemiology, but also health problems like in diabetes, metabolic syndrome, kidney and cancer. And of course also again personalized profile, monitor and optimize treatment, and look into food-related changes. So here I only show a small fraction of it, because we have here, of course, 150 metabolites in the end, and you see the same principle. You see the ones which have been identified. For each of those, the DIN isospiking has been done, but we also have a special version of what we call electronic or numerical spiking to further identify correct LODs. You see the 95% range that we have in our model data set. And you see absolute and relative concentrations relative to creatinine. Then last thing is biobanks. Of course here extensive quality control is very important to sort out aliquots with low quality, or at least to mark them that there is an issue. And of course then the QC process is actually done in quite a few minutes, like 5 minutes, and then the spectrum actually can be recorded and transferred into the biobank, and then also the analytical data, as I showed you, quantification in urine, plasma, serum, actually can be done. So I think that is what I actually wanted to present. So now actually I asked the 2 professors to begin their presentation, and we start with Professor Claudio Luchinat. Claudio, please start your presentation.
Okay, thank you, Manfred. I thought I should say who I am, so you already said that, and I may actually like to show the place where I'm working, and this is the Magnetic Resonance Center of the University of Florence. And you see the numbers are the instruments, the various NMR instruments with their frequency megahertz or field. And the ones that are circled are the ones that are used for metabolomics. In particular, one of the 600 megahertz is an IVDR system. And so we are standardized in the sense that Manfred was telling a few minutes ago. And my idea was to give you a very quick overview of the approach that we had to metabolomics, and speaking about health metabolomics, although we also look at food. And, well, this is, let's say, one of the 2 or 3 concept slides that I have. Basically, this is the whole, let's say, flow of information and strategy to obtain metabolomic data. And you see that there are several ways to do that. I'll concentrate on untargeted analysis, and perhaps I can highlight that. And as you know, untargeted analysis can be done by just looking at the spectra and fingerprinting the spectra by binning, or looking at quantifiable metabolites, and so this is called profiling. We have been doing mostly fingerprinting, and I think I'll take this opportunity to tell you what we think about the various approaches, and then move to multivariate analysis, and then we often use supervised methods to, for instance, discriminate healthy people from diseased people, and this is the opening the way to actually being able to provide early diagnosis of several diseases. This is just a few flash examples of what we have done, and this is all CERMMP. I'll concentrate on CERMMP today, where we have actually looked at diseased and controlled people. This is standard statistical approaches like multivariate analysis. This is PLS, for instance. And you see that people with the celiac disease are clearly discriminated from people who are controls, who are healthy. What was rather interesting when we published that several years ago now was that when these people were put on a gluten-free diet, which is what you usually expect a doctor would prescribe for celiac people, you see that the red dots become green and they move to the right. So they move in the field of the healthy controls. This is a little surprising because, as you may know, celiac disease causes intestinal damage, and intestinal damage may not always be reversible, but if it is taken on time, it can be partially reversible. So this tells you that indeed there is kind of normalization of functions when people are put on a gluten-free diet. Again, this is done by fingerprinting, so it's just taking the spectra, binning the spectra, and looking at the various bins or buckets to use them as variables for statistical analysis. Even more interesting is that we looked at potential celiac people, and potential celiac people are people who have the antibodies typical of celiac disease but don't have intestinal damage. So these people, we thought at the beginning that they would be normal because the way of functioning of their intestine is normal, but on the contrary, most of them were in the field of celiac disease people, which to us was very important because it signaled that indeed the metabolic profile can actually help diagnosing celiac disease before people even think of having the problem, which usually manifests as a kind of reactions to food and so on, and then it may take time. Not all people are recognized as celiac immediately, so this may have strong potential implications. Likewise, this was a complex study of which we did only a small part, which was colorectal cancer. We looked at people with metastatic colorectal cancer after third-line treatment, so people in, say, very bad shape, unfortunately. And here the accuracy of recognition of diseased people versus healthy subject is 100% because it's really the disease is well advanced, and in the end, ultimately, all of these people died by metastasis. But what was interesting and was somehow rewarding also for the physicians with whom we were collaborating was that among these people who actually died between a couple of months and a couple of years, we could actually tell or predict the lifespan by metabolomics, by looking at, again, this was serum profiles analyzed by fingerprinting. And you see that you can tell short survival people from long survival people with an hazard ratio of 3.3, which is remarkable because there is no other indicator here that is as accurate or as strong as what we are seeing by metabolomics. So in the end, this could actually help planning palliative care and ease somehow or improve the quality of life of these people, even if the disease could not be healed. Breast cancer is a series of papers that we have published over the years, where we first identified the fingerprint. This is again serum fingerprinting by bucketing. The fingerprint of breast cancer against normal people, but we also could see a clear fingerprint of metastasis against breast cancer without metastasis. And then we thought we might use this fingerprint of metastasis to try and see whether we would be able to predict recurrence or predict relapse or whatever on women that at the time of surgery had no sign of metastasis, but then after a few years they would develop metastasis. And what very interesting is to see that we had a prediction over 80%. And this is remarkable because all of these women would have been treated more or less in the same way. We could actually provide an additional hint to the physician on whether or how strong should the chemotherapy be, depending on this type of metabolic indicator of chances of getting a relapse. And this study was later validated on a larger cohort, which was very heterogeneous from different hospitals from the Philippines and Vietnam and Bangladesh and a couple of countries in Africa. And the prediction was slightly lower, but still remarkable, accuracy 73%, which was somehow telling us that the predictor is robust enough to be further tested on larger cohorts and perhaps even adopted as an additional tool in clinics. As I said, accuracy is lower, but not that bad. The last example is heart failure metabolomics, on which, again, by simple fingerprinting of serum, we could tell easily people affected by heart failure versus normal people. But the interesting thing here was that these people were actually undistinguishable in terms of stage of the disease, from very, very early to very severe. There was no way to discriminate. So it looked like there is a fingerprint of heart failure which is independent of the seriousness of the disease, independent of the evolution of the disease, and which might actually be present even before symptoms. And we checked that, and this is not published, but we checked that on a cohort of about 700, plus more than 700 blood donors, on which we looked for the fingerprint of heart failure. We found 20 of them with a fingerprint, NMR fingerprint of heart failure. We were able to get hold of 11 of these people and ask them to undergo a heart echography. And out of these 11 people, 6 had altered parameters that would indicate the beginning of heart failure. So we think, and this is not published because the numbers are too small, but we think that there is indeed the possibility that fingerprinting could become a way to early diagnosis, or at least to address these people to more specific tests. Now this is the same slide as before, and now I would like to move to profiling because this is totally different. And we are actually, I have to confess that we started doing profiling not long ago, and thanks to Brucher and thanks to IVDR. And so I would like to show you some considerations about profiling versus fingerprinting. And this is my last concept slide, where at least this is the way we see it. I mean, if you do fingerprinting, and if you do it well, all the information that is present in an NMR spectrum is collected somehow by fingerprinting. Even if you don't know where it comes from, even if you don't know the identity of the metabolites that cause one fingerprint to be different from another, but the statistical discriminating power is the highest. Whereas profiling, of course, depends. If you use the metabolites that you get from profiling as a tool to discriminate groups, then you need to have many metabolites. Now, this goes up to 1,000, but it doesn't need to be so high. But you need many metabolites in order to get asymptotically to the same amount of information that is naturally contained in fingerprinting. So our first question was, why profiling? Since we seem to be actually rather successful with fingerprinting alone. And there are many reasons. One is portability, because if you do fingerprinting, you're linked to a specific instrument, to a specific, very strict SOPs that we use. Thanks to Brucher, we are, with many other labs, we are standardizing the way we acquire the spectra, and so on. But this precludes, for instance, comparison with fingerprints taken at different NMR fields, or even with other techniques like mass spec. The other point, of course, is biological insight, because then when you know what are the metabolites that constitute your profile, or fingerprint if you want, made by a profile of metabolites, then you can learn about the biology, physiology, biology, pathology, and so on. So that is invaluable because you can learn something about the disease. And finally, profiling is less prone to artifacts than fingerprinting. If you have a strange preservative, for instance, in vials and so on, you may miss the presence of that by just not looking at the spectrum but doing fingerprinting, whereas if you look at metabolites one by one and quantify metabolites one by one, this cannot happen. So this is a great advantage. Now, you have seen this from Manfred, so I don't need to go through it. You have seen that this report is very detailed, and it is detailed. This is for lipoproteins, and also you have seen that there is a very detailed report for small metabolites, small molecules. And we have a few examples that are all in progress, all very preliminary, and nothing is published, so I cannot even give you so much details. But we have actually started using this IVDR from Bruker, this predictor. And this is discrimination between hepatitis C from hepatitis B. This was not dictated by diagnosis, but just by the curiosity, scientific curiosity to see whether this could be discriminated and also discriminated from healthy subjects. And the result is that if you look at the first panel on the left, this is what you get from the 34 metabolites that have been identified in CERMMP. I should say that these were not identified by the Bruker software because this was before, but still it's a very similar thing. And you see that the predictive accuracy among the different groups, so hepatitis C, hepatitis B, and healthy subjects, is not so high, 68%. Whereas if you go to the lower right, you see the predictive accuracy by binning, by our standard fingerprint method. It's 78.2, which is significantly better. Then what is very interesting for us now in this webinar is that if you use the lipid subfractions, which is 114 different variables, you see that the discrimination is almost as good, or practically identical to what we get from binning, which means to us that what we are capturing from the noisy spectra is probably mostly the broad signals, the lipid components, and which are clearly identified, and of course with all the extra information that you get from having identified them. You heard from Manfred that if you can tell the various sub-fractions of LDL, you can have a much more accurate diagnosis, much more accurate risk factor, and so on. So of course, for equal or almost equal accuracy, then of course profiling is much better than fingerprinting alone. And then this is a study that was a big European study, of which we got a few samples as a small side project here in Florence. And we were looking at women with breast cancer, and the collection was very large, so we were given only 192 patients who had breast cancer, because the idea was to see whether there was a difference between women with high mammographic density, which is known to be a risk factor for breast cancer, and low mammographic density. So this was a very challenging project, because, and I think it's still challenging, because maybe there isn't any difference. So we looked, and the best discrimination is given by diffusion spectra, so this is still done by fingerprinting, and the discrimination is very poor, 62%. And if we look at BLIS again, so the lipid components, we see that at least triglycerides are significantly higher in low density. So low density is lower risk, and if anything, this study would suggest that we should keep a close eye on triglyceride on women with low density breast, because that could be a risk factor that is not so important for high density women. So this is more detailed. You see all the fractions, and you see that these two, VLDL1 and IDL, are actually significantly different from the women with high density. Finally, this was a project on nutrition, which is still going, or is going to close shortly. It's a European project where we looked at people with people at risk of poverty, and whether the diet of people at risk of poverty would be different significantly if that would have an impact on health. I can tell you that the impact is not great, and sometimes people at risk of poverty eat well than wealthy people. So, but it was interesting to see that again we had a significant difference with triglycerides, and again we did the ELISA, and most of the triglycerides were significant, some barely significant, some more significant. And that seemed to be a very clear way of discriminating these groups. So the discrimination, I should have said before, it's before starting the diet and after the diet. So this was a diet which was the same for everyone. So as I said, this is a side result of the project that was aimed at identifying risk factors in people at risk of poverty. Last project, that I would like to present today is a study on colon cancer and high and low recurrence risk. And again, this is interesting just to show that, again, compare the accuracy that we get from fingerprinting with the accuracy that we get from profiling. So not considering all the extra information that we get from profiling, we have an accuracy by the 25 metabolites quantified by Bruker IVDR, an accuracy of 80%, which is significant, very significant. Of course, we have a higher accuracy in the fingerprinting, but this is obvious because, of course, you have more information here, but we are getting close. So this speaks somehow in favor of continuing the effort of identifying and quantifying more and more metabolites to get closer, asymptotically closer to the amount of information that is contained in fingerprinting, with all the extra benefits, of course. And here you can tell which are the significant, and this is very significant, a log 2 of -2 or so, 3-hydroxybutyric acid and acetone, and this was significant to identify high risk and low risk. And but it's not so relevant what we gather, and this is still a study in progress. Lipids are also informative, not as informative as diffusion spectra. You see that there is a parallel behavior. Diffusion spectra are sensitive to large molecules, and lipids, of course, are usually give broad signals, so it makes a lot of sense. So again, I think the biological conclusions I cannot give you now because this is the very beginning of the study, but it's from our point of view encouraging that if we look at the right stuff, we are not losing much of the information, and we are gaining a lot in terms of this is zooming on all the differences. Again, triglycerides seems to be important, and I think I will close here. And this is the group of people who work on metabolomics in CERMMP. There is also people who work on food that I'm not been speaking about, but the idea of quantifying as many as possible metabolites and other components, I think it's a winning idea. And with this, I thank you for your attention.
Thank you, Claudio, for this really comprehensive review on what your work is actually relating to. So I would now like to switch to Professor Elaine Holmes. Elaine, please, the stage is yours now.
Thank you, Manfred. Okay, so I'm based at Imperial College in London, and a lot of our NMR facility is part of what we have formed into the National Phenome Centre, and this is really set up to do high throughput screening of biofluids, and mainly in the clinical arena. We also have a patient or more personalised medicine approach, which is part of our clinical Phenome Centre, and together they rely on NMR and mass spectrometry, but very heavily on NMR spectroscopy. So what we really look at in our laboratory is phenomics, and by phenome what I mean is an integrated set of measurable clinical and physical variables, and we couple this information to chemical, metabolic and physiological properties that define different biological subclasses and individuality, and we're looking at this in the context of health and disease. And we also know that the gut bacteria, not just the human, but the bacteria, the viruses, all the other microorganisms within us also have metabolic processes, and we can also capture these by spectroscopy. So the idea, as you'll have learned from Manfred and Claudio, is very much that we measure biological samples, we sort of fingerprint them, and then we perform some sort of modeling on them to look for either diagnostic or prognostic information about different diseases, and we can take the biomarkers and model them using various tools. But the part we're focusing on today is the spectroscopy. So why do we use NMR spectroscopy? Every spectroscopic platform has its own strengths and weaknesses, but we are essentially coming from an NMR perspective, although we do use a lot of mass spectrometry now. Most of the founders in our laboratory started off as NMR spectroscopers because it's such a robust platform, and it delivers, it's really the only technique that delivers information on atom-centered properties. Claudio very nicely described the difference between untargeted and targeted profiling. And when we consider untargeted profiling, there's always going to be a little bit of interlaboratory variation. But having said that, NMR spectroscopy has repeatedly been shown to be reproducible and robust in a high throughput mode. So you really can use it and share your data between laboratories. So that's another attractive feature of NMR. Now, if we're thinking of targeted, whereby we model NMR data in a quantitative fashion, then the results between laboratories, which I'll show you a little bit more about later in the talk, is really strongly reproducible, and it allows that together the untargeted and targeted approaches allow both hypothesis testing and hypothesis generation in the same experiment. We also use NMR as a first-line screen to detect any outlier samples before progressing onto other analytical platforms. So even if we're going to carry out both NMR and mass spec, then what we would do is first run the samples through NMR just to make sure that the sample isn't an outlier, and by doing this you cut down on samples that would really contain high lipids or high proteins and would damage a mass spec column. So we use it as an entity in its own right to deliver scientific information, but we also use it as a first-line screening tool, and that is very important to us because it enables us to get through tens of thousands of samples a year. So we have 3 NMR spectrometers dedicated to the National Phenome Center, and if we consider a relatively easy throughput of 96 sample assays per day per instrument, which isn't full capacity, but it is an approximation to what we're currently running, we would end up running on the 3 spectrometers 288 samples a day, or over 100,000 samples per year. And these samples across the 3 spectrometers are near on identical. The 3 spectrometers give near on identical results. So sample workflow is very important. There have been various publications from various groups on this, but we basically follow Bruker's protocol, and the whole philosophy is described in an article by Dona et al. But you take your sample, you do some sort of sample manipulation and storage. When you're ready to run the sample, you then prepare the sample, and it's very important that the preparation methods are done following a very strict SOP or protocol so that you can get the same results across laboratories. Then you have the analysis itself, for which again there are protocols and procedures prescribed, and the following step is to do pattern recognition or quantification of metabolites and come up with some sort of biological hypothesis. So what we're aiming to do in the metabolic profiling community is to achieve harmonization. And whilst this is very difficult, for NMR we've almost got there. So just reiterating, we need to ensure SOPs and analytical pipelines are consistent. And we do this by sharing of SOPs and protocols and ring trials, one of which Manfred touched on briefly and I'll come back to, but also sharing of databases. So if we follow these very well-prescribed protocols, what we end up with is data that we can share. And for rare diseases, for example, this is important because we can share information on samples, real biological samples, and whilst we may not have enough in our own centres we're looking at, we can share with other centres and get a critical mass for various diseases that may otherwise not be modelled adequately. So the lipoprotein ring test was a test that we undertook between ourselves as the National Phenome Centre, the Phenome Centre in Birmingham, and Brookers. So we had three different physical located centres, and in them we have 5 different institutions because we include the Clinical Phenome Centre and the research laboratory. So within those 5 institutions we have 11 different NMR spectrometers, and we measured 2 daily quality control samples for 6 days of analysis, against 2 replicates of NIST samples, and then we looked at 40 different donor samples from real patients. Now, for a lot of the work we do for cardiovascular disease, for cancers, and just for everyday physiological variation, then lipoproteins are very important. So using Bruker's BI-LISA method, we can generate data, accurate data, quantitative data on 105 lipoprotein particles and 24 small molecules. And the next slide, the slide we're showing here, just shows you on the top panel, C, that the lipoproteins are calculated mainly from 2 regions of the NMR spectrum, which involves a complex deconvolution of those peak shapes. But from those, we managed to get accurate decomposition and values on the lipoproteins. At the same time, we measured the 24 small molecules, and you can see from the bottom 2 spectra below, these are the standards, that these molecules are overlap, they're complex, and yet we've shown that that by using the method Bruker have developed, we can get good reproducible information. So in order to develop this method, the reference cohort the samples were collected and ultracentrifugation was done to assign the lipoprotein profiles, and we compared this with those we acquired in the NMR laboratory. And Bruker conducted the quantification using an electronic signal in the spectrum to calculate the proton concentrations. Just looking at linear regression between the BI-LISA method and the clinical measurements, you can see whether it's total cholesterol, HDL cholesterol, or the apolipoproteins, that we have very high correlation between the NMR method versus the conventional clinical method. The intra-institutional reproducibility of lipoprotein concentrations was extremely high, as you can see in this slide. And if we divide that down and have a closer look across the 11 spectrometers for the triglycerides, cholesterol, the ApoA1 and ApoB, and the LDL and HDL, you can see across those 11 spectrometers, the standard deviation is given in 2 times standard deviation is given in light green, 1 times standard deviation given in dark green. You can see they're very, very tight, highly reproducible. So for us, this was an amazing result. And if you do an in-depth analysis of one sample across the 11 spectrometers, they're all superimposed on each other, color-coded. The little diamonds are color-coded according to machine and institution. You can see they're largely totally superimposed. And for some parameters, there's a tiny bit of variation. But if you take a single sample and you analyze them across the 11 institutions, you get high reproducibility for every single one of those 105 lipoprotein parameters. The same holds more or less for the low molecular weight molecules. I'm showing 6 here, 6 of the common common molecules of interest to the clinicians, so amino acids, creatinine, lactate, branched chain amino acids. And now I'd like to finish by showing you a couple of applications. So one of the things we do here is to look at pregnancy and complications of pregnancy. And before we look at complications of pregnancy, you should look at normal pregnancy. And across the total gestation period for hundreds of women, we collected samples and we ran the profiles using standard NMR spectroscopy. So these are just the profiles, it's not the quantitative metabolites here, but you can see that between the first, second, and third trimester, colored in blue, yellow, and gray, you can see the separation between the trimesters. So as a woman goes through pregnancy, unsurprisingly, her plasma composition changes. So since lipid metabolism showed the largest gestation-associated variation rather than the small molecules here, what we did was to do an additional lipoprotein subfraction distribution analysis, and we used the BILISA platform to do that. So we looked at 105 lipoprotein subfractions. If you perform univariate analysis on this, then 95 of those lipoprotein fractions are different or shift in metabolism during a healthy, uncomplicated pregnancy journey. And this isn't surprising because we know that lipid metabolism is altered in pregnancy, and I think Ruth Ley has shown that in mice, if you take third trimester urine and transplant it into other mice, then germ-free mice, then they've developed cholestasis. So every woman is in a mild state of cholestasis during pregnancy. However, you can narrow this down and get a pattern of more predictive lipoprotein profiles, and here we resorted to using 38 of the top 95, the top 38 of the 95 significantly changing lipoprotein subfractions. And what this enabled us to do was to build a quantitative diagnostic for lipoprotein changes during pregnancy. And you can see from the ROC curve here that the area under the curve is 0.94. So very high predictive coverage of gestational age. So we use this model of healthy pregnancy to be able to look at the difference between healthy pregnancy and those women who went on to deliver preterm. And the two lines in the bottom right panel there, you can see two lines in blue, the trimesters for the term babies, and in red preterm. You can see by using the healthy pregnancy model, you can see the lipoproteins for the preterms are totally different. Now switching to urine, and we're looking at exactly the same concept. So how does quantitation of urine help us? Bruker have a nice fitting method for quantitative fitting method for urine. Because the Bruker method is not published, it's obviously proprietary, we wanted to test it out against our own method. So we used Joran Posner, one of our postdocs, developed a method of curve fitting to quantify various parameters, various metabolites, and we compared it with the Bruker. Now, the Bruker method, I have to say, will be much more accurate because they put years of work into this, whereas we're comparing it with an in-house method that was developed over just a couple of months. But what we found was that if you look at the creatinine concentrations, then there really aren't that many outliers, and the two methods agree beautifully. So we looked at this across 7,500 samples, 7,500 individuals, and find incredibly high reproducibility. So knowing the creatinine is fitting well, we then moved to look at other metabolites, and I think we're looking in total of about 200 metabolites. For those that overlap with Bruker, which was 48, we found good correlation, mainly lower than 5% with a couple of metabolites that were outside that range, possibly to do with relaxation, and the methods we're using don't quite accommodate in Imperial for the overlap of peaks. But what we find is, again, good correspondence between the two methods. So one thing that's missing in the literature is a really comprehensive validation of physiological variation. Here you see gender differentiation. This is based on 6,000 individuals, and you can see from these 48, and actually only showing 17 here, but of the top 17 differential metabolites, you can see strong differentiation between males and females, and you can look at the distributions here. We can look at the same for age, and then we can start to stratify these metabolites by age and gender. So you can see for lactic acid, in females over age, the lactic acid drops in concentration in the urine, whereas for males you don't really see this effect. So we have different patterns here associated with age and gender. So for the first time we've been able to establish a really comprehensive set of reference ranges for physiological variance, looking at a wide range of populations. And you see different behavior patterns for different metabolites. So alanine, there's only a weak effect with age and gender, whereas for other metabolites like dimethylamine, you see both an age and a gender effect. So what we've done is to really comprehensively classify distributions of metabolites over age, and we're using these now for our pathology studies, so we now know what physiological variance looks like, and we can translate this into our disease-based studies. So to summarize, what I hope I've shown you is that the accurate quantification of lipoproteins and small molecules in plasma and serum is possible using the BI-LISA fitting method. These quantified metabolites can be used to form biomarker panels for prediction of physiological and pathological states, and is highly suited to high-throughput profiling. Clinicians, we found, don't like profiling data because they don't understand it, so what they want is a set of quantification or quantified metabolite values, and this is what we can now deliver to them. I showed you a couple of examples, one in healthy pregnancy, showing you the vast change in lipoproteins and how we can use this to model healthy pregnancy and to predict unhealthy pregnancy or adverse outcomes in pregnancy. Similarly for urine, it's consistent, the Bruker quantification is consistent with other peak fitting methods, and we've now ascertained a range of metabolite concentrations which we're now using and employing in various disease-based studies. So finally, I'd just like to acknowledge the lab members who have either helped me produce the slides or worked on the quantification algorithms with us, and our clinicians for providing the pregnancy study data. So thank you for listening. I'll hand back over to Manfred.
Thank you, Elaine. Very informative presentation. Also, thank you again to Claudio. Now I talk to the audience. If you have a question, we have received quite a few questions already that you want to ask, then please do so now. You can click on the answer a question box, which is located on the far left of your screen. We will answer as many of your questions as we have time for. The ones we can't answer, we will actually then answer by email later after the webinar is finished. So I want to get started with the questions. So I start questions for the lady first. Elaine, I have a question now. What is the balance between untargeted and targeted analysis in your laboratory?
So when we started out, it was 100% untargeted analysis, and personally as a scientist I would always prefer untargeted analysis to start with, because it allows you to do true exploration and find molecules or biomarkers that you don't know about, so it gives you the exploratory capacity. However, as I kind of alluded to, our clinicians really weren't so happy with the profiles because they weren't equipped to be able to look at the profiles themselves, and what they really want is an easy answer. You know, which biomarkers can you give us metabolites that are diagnostic for a particular disease condition, and what concentrations are those metabolites in? So now we find that there's a big demand from our clinicians to have annotated data. So I think this is increasingly becoming more and more important. And I would say 50% of our studies now we do full annotation on.
Thank you, Elaine. So then the next question actually goes now to Claudio Luchinat. Question is there in the EPIC study, is there an explanation for the increase in triglycerides? And also, since also the small molecules are available now, is there also an effect of the diet that could be studied on the small molecules?
Yeah, so the first part of the question is that no, we don't have an explanation for that, but we seem to observe that women that got cancer with low density, they were having higher values of triglycerides, which might mean that, and this is not related to diet. I mean, this is an intrinsic thing that discriminates, not 100%, but anyway, discriminates women with low density, as if let's say the composition of the lipid composition was more important for them than for those with high density. As I said, this was a kind of very, very small side thing within the big EPIC project, and we are still working on that. So these are the first data that we got, and our collaborator in Florence was kind of excited at seeing that this was the first evidence of a difference that they could tell, besides the density itself. So from here we could try to go and look at what you were mentioning, the effect of diet on that. Of course, small metabolites would be also meaningful, and we had already started looking at them, let's say by hand, before starting using your program. So we will have to do it again with your program, and hopefully also to identify more than 20 metabolites. I mean, this is already a good number, but I think what we learn from this, what we are learning, is that the higher the number of metabolites, the closer we get to the full panel that would be equivalent to fingerprinting. And I think, as Elaine was mentioning, I think it's a matter of culture also from the medical community, that they should start realizing that a panel of metabolites is a marker, not just one metabolite, but a panel can be collectively considered a marker.
Okay, thank you, Claudio. Now back to Elaine. Question about the pre-analytics. How important is it to stick to the protocols for sample collection and preparation?
This is absolutely essential. I'll start by saying that again, NMR is a little more forgiving than mass spec. However, if you want to get good quantification, then we've found that really, you need to follow this prescribed protocols, and sample collection in particular is very important. How quickly you store the serum, how long anything is left on the bench. This all impacts on the profile. If you have the same samples that you distribute across sites, it works fine, and largely you can correct for center effects, but it's much better not to have to do that. Now, there are instances, for example, it's better to collect 24-hour urine samples, but this is usually highly impractical in a clinical setting, and you end up collecting first morning urine, or often even not that, maybe it's spot urines throughout the day. So there has to be a little give and take in the system to accommodate the practicalities of the current clinical practice. But where possible, it's absolutely, the sample collection should be followed, and the protocols is absolutely essential to follow them thoroughly.
Thank you. So back to Claudio. Next question, how important is the standardization in NMR for your research? How does this compare to other analytical methods you are using?
Okay, I'm happy to answer this question. I think it's vital. If you don't standardize, you will never get anything meaningful. But as it was already pointed out both by you and by Elaine, NMR is a powerful tool, because if everything else is done properly, from the pre-analytic procedure to the analytic procedure to the preparation of the sample for NMR itself and the way the spectra are acquired, NMR is probably the best technique in terms of quantification, reproducibility, and so on and so forth. I mean, we don't have such a great experience with other analytical methods, but I know enough to say that usually analytical methods have a larger, even the good ones, have a larger uncertainty than what you would get from quantitation of NMR signals if it is done properly and if the sample is prepared properly. I think the bottleneck is the absolute reproducibility in the preparation of samples.
Yeah, thank you. Now I also got a question actually to my part, and the question is why is there a difference in quantification between newborns and older children and adults? Well, the reason for that is that in the early days of life, actually the ionic matrix, which the urine represents, is quite different from the matrix that you have when actually children are aging, turning into adults. And of course, this difference in the ionic matrix will be salts, for example, that actually influences the chemical shift of the metabolites in the NMR spectrum. That means the position of the metabolites in the NMR spectrum. And in order to have the best sensitivity and specificity in this identification and quantification of metabolites, we actually have separated those, and this leads to actually lower LODs, as if we would go over the whole range, and of course then also much higher certainty in identification and quantification. So, okay, I think there is just time for one more question, and I put that to Elaine. How do you see these quantification tools impacting on clinical practice?
I think now the age of precision medicine and also biobanking and high throughput screening has put us in a very good position. The technology and tools we have here will really serve the clinicians well because we can do simultaneous measurements on numerous metabolites. So I think now is the perfect time to marry metabolic profiling with real clinical practice. So I think it's perfect timing, and the impact on clinicians to be able to process all this extra information to move towards better disease risk markers and better methods for precision and personalized medicine. It's so important, and we're right there now.
Okay, thank you, Elaine. Thank you, Claudio. This was really informative information that you have presented, and unfortunately we couldn't actually answer all questions, and we are still receiving questions. So as I mentioned before, these questions will be answered by email. And now I think I give the ball back to Christy to finalize the webinar.
Thank you, Manfred. Now, before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. Additionally, a short exit poll will pop up on your screen. Your feedback is important to us, and we thank you for participating. We would like to thank Professor Luchinat and Professor Holmes for their time today and for their important research. We would also like to thank LabRoots, Manfred Sproul, and our sponsor, Bruker, for underwriting today's educational webcast. This webcast can be viewed on demand through February 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We hope to see you again soon. Goodbye.